I'm Daniel Levine, and this is the Bio Report.
The advent of immunotherapies is transforming cancer care. While early efforts have focused on enabling T cells to attack cancers, Myeloid Therapeutics is developing next-generation immunotherapies that use mRNA to train myeloid cells to attack cancer. We spoke to Daniel Getz, founder and CEO of Myeloid Therapeutics, about the limitations of existing immunotherapies, the role myeloid cells play in the immune system, and the benefits of the company's therapeutic approach. Dan, thanks for joining us.
Danny, it's a pleasure to be here, and I really appreciate the opportunity to share some of the amazing things that we've been working on.
Well, we're going to talk about mRNA immunotherapy, myeloid, and the benefits of its approach to treating solid tumors. Let's start with the state of immunotherapy today and the limitations of current approaches. How successful have they been, and what limitations have we seen to date?
Immunotherapies like checkpoint inhibitors and CAR T cells have certainly transformed cancer treatment, especially in blood cancers. But their successes in solid tumors has been far more limited. If we look at some of the statistics as an example, 5-year survival for solid tumors for most of them as a group is still less than 50% and much less in areas like hepatocellular carcinoma or in pancreatic cancer. Also, checkpoint inhibitors generally are not effective in more than 70% of solid tumors. So we still have a long way to go in solving solid tumors. And we believe that the key to overcoming this is really addressing the tumor itself and its environment, which really suppresses immune activity. So that's really where the focus has been. And whilst the immunotherapies we have today have certainly changed the game, we really need to focus on solid tumors and for those patients that haven't benefited from those therapies that are out there today.
A lot of the early focus and success of immunotherapies have been by targeting T cells. Your company's targeting myeloid cells. What are myeloid cells, and what role do they play in the immune response?
Yeah, so myeloid cells are an amazing population of cells. They are actually the immune system's first responders and orchestrators of immune responses. They detect threats, and they are the first, if you like, responders to run into the burning building. If you have a infection, for example, or a traumatic injury, these are the first cells on the scene, and their primary job at that point is really to wall off, for example, a pathogen or limit any further, damage that a trauma may have. But they also then serve as a key bridge between that innate immune response and the broader activity in the adaptive immune system, such as T cells as well as NK cells. And they serve to an important function in activating T cells because they actually present what we call antigens to those T cells to make them become active. So to have a fully effective T cell response, you must actually have a myeloid cell component to it in the context of those myeloid cells essentially instructing the T cells what to do. And unfortunately, in cancer, the tumor often hijacks the myeloid cells, to suppress immunity. And so we're very much focused on trying to address how we take these first responders that are commonly found in tumors and get them to switch back on and drive this integrated immune response.
How robust an immune response are they capable of eliciting? And do they end up activating other immune cells beyond T cells?
Yeah, it's a great question. So, what's really interesting in my background actually is in how these myeloid cells originally, during my PhD, cause immune pathology. So, these myeloid cells can actually become extremely activated. They're a primary cause of a lot of pathology in some disorders like SARS syndrome, for example, in the lung, severe acute respiratory syndrome. They can also cause in virus infection of the brain pathology there. And this is really all driven by their potent production of inflammatory cytokines, their ability to attract other cells to the site of inflammation. So these cells actually have significant artillery and can really drive, if you're not careful, a very, very potent immune response. Now, this is just through basic mediators that they can produce. But you did actually just mention, their ability to activate other immune cells, and that's really an important element to it because these cells actually produce key signaling molecules that are essential for not only activating T cells and NK cells, but they're really, really important in attracting T cells and NK cells to sites of inflammation. And in the context of cancer, this is really important because in cancers like pancreatic cancer, ovarian, glioblastoma, some of the really, really difficult cancers to treat, one of the challenges in immunotherapy is getting T cells into these tumors because they really don't let them in. And then, secondarily, the other issue is the immune suppressive environment. And what we now know using myeloid targeting approaches is you can actually get those cells to be turned on, become activated, become inflammatory, and recruit those cells there. So, in the context of biology, these cells have quite an array of tools to be potent effector cells. Uh, and, it's a really systems biology approach when you activate them because of their ability to recruit other cell types and activate them as well.
mRNA came to widespread attention with the COVID pandemic and the vaccines that were produced to combat it. Did these provide any type of a proof of concept for what you're doing?
Absolutely. I mean, RNA is involved in all cell processes. I'm sure people have heard of DNA as well, which holds the instruction for the cell. RNA turns these instructions into proteins. And of course, COVID really put RNA on the map in the context of COVID vaccines. But we have been working on RNA for, close to 60 or 70 years. And so there has been, a significant amount of research using RNA before COVID. But obviously, COVID, was really important for a number of things. It really validated the ability to harness this modality in a therapeutic setting. It showed that we could safely deliver and rapidly manufacture this therapeutic. And it also showed we could use it to program immune responses in the context of generating, predominantly B cell responses, antibody responses against, a viral antigen. And so I think in the context of RNA therapeutics, COVID was absolutely amazing and really paving the way. And I think when we think about the fact that I think it was about 11 months from the day they started working on the COVID vaccine till its approval, and you think about the hundreds of millions of patients that have received COVID vaccine dosing, it's obviously been shown to be relatively safe. And I understand there's controversy around that. The controversy is questionable in my mind. Very few therapies have the same sample size of being applied globally across hundreds of millions of people and showing a safety profile that essentially is really quite unparalleled, in my opinion.
One of the compelling things you're doing is developing mRNA-based immunotherapies that program immune cells in vivo to combat cancer. You have two clinical programs that are using lipid nanoparticles as vectors. What are the elements that make up a therapy? What's packaged inside that LNP?
So at Myeloid, our primary focus has been on how we train our own immune systems to recognize cancer and elicit an endogenous immune response, your own immune response against it. So our product is comprised of a nanoparticle, a lipid nanoparticle, which is very similar, obviously, to what is in the COVID vaccine. A primary difference or an important element of it is the mRNA sequence inside um, the LNP. And that essentially is the code for a protein that, when taken up by these immune cells, they make a protein that is expressed on the surface. And it is essentially a homing protein that allows the myeloid cell to see the tumor and then become activated. And the reason why this is important in cancer is I mentioned earlier that myeloid cells are the first responders in trauma and in viral infection. Well, in those situations, there are signals from damaged tissue or from the virus that activate the myeloid cell. Now, in the context of tumors, the myeloid cell is not able to actually recognize the tumor as being a problem because it sees it as kind of self. And what we've done is, by delivering our RNA coding for this protein, that protein serves as a bridge between that cell and the tumor, and it tells the myeloid cell, hey, this is not supposed to be here. It becomes activated, and then it causes that cascade of events that result in direct killing of the tumor as well as integration of this broad immune response.
Is there any concern about potential off target effects?
It's a great question as well. Dennis, you're probably quite aware with CAR T cells, there's often been what we call on target off tumor side effects. So these are essentially situations where the cell recognizes the target, but that target's also expressed, although often at a lower level, on healthy tissues. Now, what's really quite different between CAR T cells and this myeloid approach is T cells have an ability to essentially survey the entire body. They're able to migrate between tissues, they can go into the brain, and so they can search out, if you like, these targets. Now, that means that if there's a low expressed target, for example, in the brain, that T cell could effectively go in there and become activated when it sees it, even though that's not a tumor target necessarily. So, what's really different for us is that myeloid cells, 99.9% of the time, can only be called into a site whereby there is a molecule calling them there. And what's really quite interesting in cancer is cancer actually makes a lot of these, what we call chemokines, and it attracts those cells in there. So we have actually dosed now around 30 patients across our programs, and we haven't actually observed any what we call on target off tumor side effects. The side effect profile that we are observing is one that is really akin to immune activation and an actual mechanism of tumor killing. And we can get into that in more detail, if you like, in a moment.
You talked about the problem of the tumor microenvironment muting the myeloid cells. How do you get around that problem? Why doesn't that happen here if it doesn't?
That's a great question. So the protein that we are delivering to these myeloid cells incorporates a signaling domain on it that actually activates the myeloid cell upon recognition of the tumor and overcomes the tumor suppressive signals that are being imparted by the tumor microenvironment. So we're able to essentially overcome the suppression, and then that obviously causes a cascade of events. And we've actually seen this and have shown this in humans, looking at biopsies, pre and post treatment biopsies. When we look at the pre treatment biopsy and we look at things associated with immune activity, these biopsies appear to be what we call very cold. Not very many T cells in there. there isn't any inflammatory mediators being produced. And then we compare that 30 days later, and what we find is a very different scene, what we call a very hot tumor microenvironment. We actually see significant influx of immune cells like T cells and the production of a number of really potent inflammatory mediators that we are quite aware of overcome that suppressive tumor microenvironment. So, in the context of dogmatically shifting the TME, as we call it, the tumor microenvironment, the early data we have really is exciting.
And how about in terms of manufacturing these therapies? Are there cost and scalability advantages uh, compared to CAR T type therapies or other cell and gene therapies?
It's not even comparable. I mean, one of the benefits of the COVID vaccine, as you could imagine, is its scale up globally and its deployment globally. So supply chains are there. And you can appreciate that the scale of this has made all of these RNA products quite effective from a manufacturing point of view to make. So we are talking, you know, several hundred, low thousands of dollars to make a dose compared to CAR T cells, which cost anywhere from 150 to $200,000 a dose. These are also off the shelf products. They're administered IV over 30 to 60 minutes. So, in the context of manufacturing and cost, I truly believe that these RNA therapies, including ours, are going to completely transform medicine and bring back, I think, the pharmaceutical business model where, you know, being able to deploy these therapies globally is not only possible, but within a business model that makes it both accessible globally to patients that are in lower economic regions of the world, simply because we're able to have a model where the cost of goods and obviously the price paid for these medicines is all within a reasonable approach. And I think this, when we get into the conversation later on retrotransposons, that also applies, right? These gene therapies are costing millions and millions of dollars. RNA is going to be able to see this come down to, you know, cost of goods in the low thousands of dollars. Completely transformative.
You have two early stage clinical programs. The first is MT302, which targets colon, lung, and breast cancers that express TROP2. A number of antibody drug conjugates are targeting TROP2, and they've either been approved or in development. How effective are these, and how much of an unmet need is there for therapies that can target these cancers?
Well, the unmet need is significant. I mean, if we think about it, a lot of these drugs are approved off several months of progression-free survival, 3 to 6 months, right? So, success in solid tumors is often measured in months. And if you're someone with a solid tumor, of course, any extension of life you get is blessing. But at the same time, as a scientist and as someone that wants to really transform cancer therapy, I want to be measuring success in years, multiple of years. So the targeting TROP2 with ADCs like Trodelvy and Dato-DXd certainly has helped support the ADC boom that we're observing. But I think it's also important to remember that these ADCs are really chemotherapy on a stick, so more directed chemotherapy, and success is being measured in months, in most cases, of progression-free survival. So they still come with toxicities, and we still are not curing cancer. And obviously, this is important. And so we want to continue to push that forward. In the context of TROP2, there's another really interesting element to it, and that is that there is an ADC called Enhertu, and this targets a different protein called HER2. But that ADC uses the same, if you like, chemical that the TROP2 ADCs target. And unfortunately for the TROP2 ADCs, if you've had that product, generally speaking, you become your tumor will become resistant to the chemical and not so much the target. So as the future progresses here, the TROP2 ADCs may find their efficacy start to wane even more, simply because patients go on a different agent that makes their tumor less susceptible to it. So in the context of TROP2 targeting, our approach, I believe, apart from being a completely different mechanism, our focus is on enabling your immune system to see the cancer and kill it. It has nothing to do with chemotherapy. But really importantly, it's still going to be effective in those patients that have potentially taken this HER2 product. Their tumor has become resistant to the drug conjugate, which would then make them less amenable to taking TROP2 ADC. So I think the unmet need for us is twofold. One is we still need agents that can solve the immune problem, the TME and activation, which ADCs do not. And secondly, unfortunately, the TROP2 space, I think the ADCs there, unless a new drug conjugate was to come into play, are going to become less effective over time.
And what's known about 302 from the studies you've done to date?
So we're actually the first people to show the ability to deliver mRNA CARs in vivo. We have shown immune cells take up these LNPs. We've shown their ability to express the CAR, and we have found in tumors armed myeloid cells, so CAR expressing myeloid cells surrounding tumor cells. And we've actually been able to, we have a patient that has responded for quite some time, more than a year. They've had over 18 doses of the therapy. So if we take a step back, quite a number of hurdles have been concerning to folks using RNA therapies. One has been the ability to repeat dose intravenously these products. This particular patient I'm talking about has had more than 18 infusions relatively safely. The side effect profile has actually been strikingly promising. The administration of these products hasn't been associated with any kind of infusion reactions that so far that we've been observing, and we're finding the CAR expressing cells in the tumor. So what we have really uncovered here, in my opinion, when it comes to in vivo programming of immune cells, it's not a function at this point of is this possible? It is now a function of when we deploy this in the most appropriate fashion in the right combinations to really drive long-term outcomes for patients.
And what's the development path forward?
So for 302, we're continuing to dose escalate. So, we are really at the front edge of this program, and we haven't, landed necessarily on the top dose that we can apply. So I think that's exciting because we're seeing activity, even though we haven't necessarily got to the optimum dose. We're also now moving, because of the safety profile, it really can be applied with a number of different therapies. As an example, it can be applied with T-cell therapies. It can be applied with chemotherapy. And so this year, with some collaborating physicians in Australia and Amsterdam, we're actually moving into frontline therapy, testing it in a number of different indications, tumor types, with a number of different agents, looking to see if we can't really drive home broader immune activity whilst being used in combination with other things like checkpoints and chemotherapy and even ADCs for that matter. So this year we'll complete our phase 1 with the program whilst initiating these studies for MT302.
You're also developing an experimental therapy that targets GPC3 positive hepatocellular cancer. How big a need is there for a GPC3 positive hepatocellular cancer treatment?
So hepatocellular carcinoma is a significant issue. It's the third fastest growing tumor type, third largest killer of folks in the world that develop cancer. And to put it into perspective, if you're a patient that is diagnosed with hepatocellular carcinoma today, you'll go on to frontline therapy, which is only around 30 to 35% effective. And when I mean effective, I don't mean killing the tumor and having what we call a complete response. I mean generally may shrink the tumor, 30 or so percent over a 3 to 6 month period. Now, after that 3 to 6 month period, if you relapse, you'll go into second line therapy. And unfortunately, the statistics are less than 15% of patients that will respond to that. So, generally speaking, 75% of patients won't respond to any therapy at all. And this is one of those unfortunate tumors where, you know, life expectancy is measured, months to maybe year and a half in many cases. So the need is very, very, very significant. And treatments are really, really limited, unfortunately.
What is MT302 and how does it work?
So both our products, MT302 and MT303, function exactly the same way. They just use a different homing molecule, if you like, to recognize two different proteins. So MT302 recognizes TROP2, which is expressed on the tumors you mentioned, whereas MT303 recognizes GPC3, glypican-3. And glypican-3 is actually a protein that's mostly expressed during embryonic development. It's disappears during life, but for whatever reason, we find it commonly expressed on hepatocellular carcinoma. So it's exactly the same molecule between these programs. The big difference is that 303 recognizes glypican-3, commonly found on hepatocellular carcinoma. And so when when the myeloid cell expresses this protein, it recognizes that, and it will obviously become activated and then again drive the same kind of downstream immune responses and anti-tumor activity that the other program I mentioned earlier does.
And what's known about 303 from studies you've done to date?
So we're very excited about where we are with the liver program with MT303. So we have encouraging tolerability and anti-tumor activity so far. The very first patient that we treated in that clinical trial had a 57% lesion area reduction at 50 days post-treatment. And this is a very stereotypical, if you like, or prototypical HCC patient. Upon subsequent scans, there was some progression in the patient. However, those target lesions that were there originally remained in response. So we're very excited. Our initial observations of efficacy, generally well tolerated, and the data that we've seen thus far actually has supported us moving rapidly into frontline therapy, which we will be doing in the next month or two. So we'll have essentially a third line study going on. The one that's happening now is monotherapy. And we'll also be moving a first line therapy with our product in the current standard of care.
Dan, beyond liquid nanoparticle vectors, you're working on an mRNA gene editing platform. Can you explain what a retrotransposon is and how these can be used as therapeutic vectors?
Yeah, definitely. So if we think about gene editing today and gene delivery, you're probably aware of approaches that are being deployed in sickle cell disease and other areas, including CAR T cells. And these use complicated vectors or non-specific delivery mechanisms that are based off viruses. What a retrotransposon is is a mobile genetic element, and it's nature's own way of inserting genetic material into DNA using mRNA as a template. What we're doing is we're actually harnessing a human retrotransposon named line 1 to deliver large gene payloads directly into the genome using mRNA. So it's non viral, highly programmable editing with the potential to really revolutionize treatments in rare diseases, oncology, and beyond.
And what might make a good candidate for a therapy, a gene editing therapy, as opposed to an mRNA therapy?
Right. So, this is also a really good question. So, if you need a durable or permanent correction, like fixing a genetic mutation, or wanting a T cell to be stably, transduced for a long period of time, gene editing and gene delivery is the way to go. For immune activation or transient control, mRNA is ideal. It's really about matching the tool to the task that you're trying to achieve.
Are you looking at this as a potential for therapies beyond cancer and at genetic-based diseases?
Yeah, so one of the really unique elements of the line one retrotransposon is it overcomes all of the limitations of size. So today, the pioneers in gene editing at Beam and Prime Medicine and CRISPR, they really are limited to making either one or two nucleotide changes within our genome, or delivering small pieces of DNA. The retrotransposon will allow us to deliver large genes at once. So, as an example, cystic fibrosis as a disorder is actually a group of essentially disorders across patients. Patients have mutations at different areas um, within their CFTR gene, and the approach that some folks like Prime are taking is trying to go in there and for each patient essentially make a personalized therapy to change that. What the retrotransposon would allow us to do is actually go into each patient and use exactly the same therapy and correct the entire thing by inserting the entire gene. So, with our retrotransposon technology, Myeloid Therapeutics, the company, we are focused on its application with oncology, but we are actively engaged and talking with partners about how to deploy this technology into those other areas, to really make gene editing and gene delivery a tool that's widely accessible and really address the full gamut of disorders that it can.
The company raised 125 million to date. Given the novelty of what you're doing, how difficult was it to convince investors that targeting myeloid cells was a viable approach, particularly given the success that CAR T cell therapies have had?
I've been very fortunate finding a group of investors that truly believe in what we're doing. And I think with any novel therapy, there's often the naysayers and those that don't believe. But I think where we sit today, now that we're able to show the in vivo safety data in humans, we're able to show the ability to repeat dose. We have CAR expression, we have immune activation. The conversations certainly have changed, and I think there are actually quite a number of investors starting to really see the potential in what we're doing. And this also expands to, strategic partners within big pharma as well, really starting to appreciate what we're doing.
Given the funding environment, has it required you to do anything different than you would have liked regarding the building of your pipeline, the staffing of the company, or how quickly you're able to bring new programs into the clinic? Has it placed a need for an emphasis on capital efficiency?
Yeah, I mean, capital efficiency is something, irrespective of the macro environment, that I take very seriously. I mean, we have brought three novel products to the clinic, and we still have quite a bit of cash in the bank to get us through some more readouts clinically. So I think being disciplined is just good business practice. um, but I think really the truth of the matter is, in the current environment there is even more pressure on making sure we're focused on that. And most recently, we have made structural changes to our business. I mentioned earlier that the safety data and the efficacy data we observed in our liver cancer product really opened the door for us to move into frontline therapy. And that effectively doubled the size of the clinical trial. So we're talking multi millions of dollars needed to solve for that. And we had to make some of the tough decisions around what we're going to cut. Now, this is always hard, especially for a company like us. Our platforms are extremely versatile. We're focused on oncology, but this same approach could be applied to autoimmune disease. Our retrotransposons, we've already touched on, have a lot of opportunities. And so, there are rationalizations and focusing that we have to do. And there are certainly things that have had to be cut down upon, because we have to double down on the areas that we think have strategic upside, and will make sure that in the future we're able to come back to those other opportunities and support them financially in better days from a macro point of view.
And how far will existing cash take you? And what's the plan for raising additional capital?
The data will always drive our next step. We're confident that, we're building something really differentiated and durable. Our current cash takes us through key inflection points in our lead clinical programs, and we continue to have conversations with private and public market opportunities in the context of the next raise. So there's no gun to our head today, but early next year we'll obviously be reevaluating our position and looking at the markets and considering capital needs at that point.
Daniel Getz, founder and CEO of Myeloid Therapeutics. Daniel, thanks so much for your time today.
Danny, I really appreciated it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.